was not really addressable until about 10 years ago because just the technology didn't exist, because these are such a tiny population of cells so deep in the brain. So one of the very first experiments we did in my lab was to investigate that, to ask for the first time what happens to these AGRP neurons when an animal eats. And so one of my first graduate students, Yiming Chen, he used a technology called fiber photometry, which allows us to put a fiber optic into the mouse's brain. So then we could record fluorescence from these AGRP neurons, which we could use as a readout of their activity. Measuring the response of these AGRP neurons was that what the neurons were doing was predicting. The mouse looks at the food, it looks at how palatable it is, imagines how hungry the mouse is, how accessible it is. And then within a few seconds, these neurons predict how much food the mouse is going to eat in the forthcoming meal. And so essentially, these neurons know how much the mouse is going to eat before the mouse even takes the first bite.